Hi, good afternoon everyone. I hope you are enjoying this community congress as much as I am. My name is Marco Hernández Valenzuela from the Instituto Politécnico Nacional de Mexico, and today I have the pleasure of being co-moderator of the session, Expansion Beyond the Limits of Resolution. Now, I would like to hand it over to my colleague, Natalie, who is going to give us a brief introduction to the topic.
Thank you, Marco. Thank you for agreeing to be my co-moderator for this session. Great to have you here. So, my name is Natalie. I'm a McGill PhD student working on expansion microscopy and STED microscopy, but my project really started with expansion, so I'm going to just give a brief introduction before our speakers get into the deep cutting-edge technology. So expansion microscopy really transformed how we image at the nanoscale, not by refining the microscope itself, but by physically enlarging the sample. So this deceptively simple idea has opened an entirely new field and new avenues for accessing molecular detail using standard microscopes, and it was developed by Dr. Ed Boyden and his team in 2015. So briefly, the core principle of expansion involves embedding a biological specimen into a hydrogel polymer and then isotropically expanding it using water. So this decrowds nanoscale structures to enable the imaging beyond the diffraction limit. And so over the past decade, expansion microscopy has evolved into a very flexible toolkit. It can be paired with STED, SIM, single molecule, and we did hear a little snippets of the stuff that Leon, our first speaker, has worked on with the other speaker in the session. And so this can be applied to diverse types of sample types and from single cells to whole brains too. So crucially, expansion helps to democratize access to nanoscale imaging by shifting resolution gain from hardware to chemistry. And it reduces reliance on prohibitively expensive instrumentation, making it a more democratized and sustainable scalable imaging ecosystem. And so in today's session, we'll be hearing from these researchers who are pushing the boundaries of what expansion microscopy can do through novel chemistries, hybrid techniques, and applications that challenge our current imaging paradigms. So these innovations reflect how expansion microscopy is not only here to stay, but continues to expand. So just a note, for our session, we'll have all the speakers come up at the front at the end for a Q&A, and so please save your questions for the very end. So without further ado, I'll introduce our first speaker. It's my pleasure to invite Dr. Yongxin Leon Zhao, who is an associate professor of biological sciences at Carnegie Mellon University. He is an interdisciplinary scientist and engineer, and his work focuses on developing tools at the intersection of protein engineering, neuroscience, and optics. His lab pioneers expansion platforms, and a major goal is to make nanoscale imaging globally accessible. Please join me in welcoming Leon.
Share screen first. Sorry, let me share screen first. Yeah. Oh, I see. Here. So everyone can see right in the Zoom? Awesome. Great. Okay, good afternoon, everyone. Thank you so much for the kind introduction, and thank you so much for all the members of the organizing committee for the invitation. It's great to be here and see all the old friends and also making the new ones here. And today I'm going to share some of the recent progress of expansion microscopy, specifically magnified, developed in our lab. So, I did not realize that actually I think I can hide this thing. Is it like this one? This one? Okay, great. I didn't know that Natalie already gave a crash course of expansion microscopy. So I'm going to go this very fast, right? We all know that confocal microscopy is diffraction limited, and then we have this super resolution find my cursor. Yeah, and but we all know super resolution systems oftentimes require very expensive equipment, complex sample preparation, and sometimes kind of slow a little bit, right? So I think the take-home message is that in order to extract nanoscale information from complex biological system, it comes as cost. But is there a way that we can think of an out-of-box idea that we'll be able to actually get this kind of information with a lower cost? So as you can guess, I mean, expansion is one of the answer. And so this technique, the concept has first coined by, my former postdoc mentor, Professor Ed Boyden at MIT, the first signature paper published in Science in 2015. And basically, you can summarize the expansion microscopy step into 4. First, anchoring, and basically, it's a chemical approach to modify the molecules inside the tissue cells, and then followed by gelling in situ, so that they form the hydrogel inside, and then after that, you apply enzyme and also expand, right? And then I can show you an animation. I steal that from Ed's talk. So basically, you have this tissue molecule with this small chemical approach them and react with them. So they have this chemical handle. With these handles, the tissue or the cell sample are submerged into the gelling solution. You can see like countless of these monomer molecules. They form this polymer chain, and during this process, the modified biomolecules will actually covalently link to the gigantic hydrogel network within. And after that, you have to go through this so-called softening process. Basically, you can apply enzyme to specifically cleave off the proteins, or you can try the heat denaturation approach to loosen up the tissue so that the hydrogel will be able to expand in the water and then separate all these proteins or other biomolecules. And as you see, in order to adapt this approach, it's a chemical approach, right? So there's some, you need to have some understanding about the chemistry. For example, if you want to do proteins, people use this direct protein anchoring approach. For example, GBIxBINA using this so-called agax, it's small chemicals. And there's other approach, for example, like using NHS or using the MAP approach. And then you have the nucleic acid that require totally different chemistry. But as you can see, there's a lot of different tricks in order to anchor those biomolecules inside the gel, and there are dozens of this technology there. The goal of my lab is try to translate this expansion microscopy technology into clinics eventually. All this actually becomes a little bit problem. And there's also other problem too. So for the classical expansion microscopy methods, oftentimes they require several days processing. And for those who already have experience working with expansion microscopy, probably you have some experience like sucking this gel into the pipette tips, or broke the cells, broke the gels. And also, as I said, like the chemistry is complicated. And oftentimes you have to look up the literature to figure out which one is the best for you, so there's no uniform anchoring mechanism there. And most of the expansion that people use 4 times expanded linearly, which is good. I mean, basically for the confocal, you can bring down the resolution to roughly 17 nanometers. However, you cannot resolve all the fine structure that you want to solve. And then you also have the limited sample capability. For example, mainly like they expand mouse brain or culture cells. And then, of course, like the enzymes, right? You chop off all the epitopes. So that means that after expand, you finish the imaging, and that's it. You cannot do anything else because all the epitopes are lost. Actually, only the labels are kept inside the gel. So my question is, can we actually figure out some expansion microscopy method that even my son can do it easily, and then also overcome all these problems? So the answer is Magnify. It's an 11-time expansion microscopy method with a universal molecular anchor. Actually, we eliminate the anchoring step. So the secret sauce is actually the methacholine, which is a molecule over there on the left side, on the right side. And it's compatible with the monomer solution of the gelling solution. So you can mix all of them together and then put the tissue inside the gelling solution, and then the anchoring and gelling process will happen simultaneously. And after that, we also developed the heat degradation approach to keep all the biomolecule in the hydrogel, and then we will expand 11 times. So I'm going to go through all these features of Magnify. Actually, after several years for development, now as it stands, the Magnify protocol already becomes a one-day process. So all you need is just a pressure cooker that you can buy from Amazon. And then you have follow this instruction, right? Basically, you have the tissue, you put the gelling solution, mix them together, wait a little bit, gel it 1 hour, and then you cut out the gel, put them in the denaturing solution with the pressure cooker, and then after 1.5 hours maximum, and you can do the washing and staining with your antibodies. And after that, you can ready for expansion and imaging. So everything combined, if you started from the morning, you finish in the afternoon. Magnify has a universal anchoring approach. So basically, it anchors all kinds of different biomolecules, for example, proteins, DNA, cyanase, and lipids. The lipid one is quite interesting. So this one is surprising, right? Because we use the surfactant, right? SDS, really harsh thing, and then you heat it up, right? And even after, like, in this case, we use 80 degrees. So that's why you have 24 hours cooking. After 24 hours at 80 degrees, you still have like 50% of the lipid contents inside the gel. So I'm just showing you like one of the images in the middle. That's the presynaptic compartment. The yellow is the synaptic vesicles, and then the lipid stain. We stain with, for example, the IDID. You can see the purple one. There's a round shape stuff there. That's the mitochondria. Actually, if you take a closer look, you can see the crystalline structures, highlighted by the lipid stain. So you can use this, like for example, in expanded human-derived organoids. This is a lung organoid, so you have some mucus secreting cells with the EVs, and then you can actually image the 3D clusters of the EVs secreted by that cells, and then you can take one of them, and with the confocal, you can do the optical sectioning, and then be able to actually reveal the complex structure inside the EV. Okay, so 11-time expansion can give you roughly 15 nanometers resolution, depending what kind of microscope objective you use. So in this case, right, just showing you, the proof of 11-time expansion on the left. On the right, we try to image synapse, right? And the panel 4 shows the EM images of synapse. And then we are looking at the structure as a presynaptic dense projection. You can see that they look really alike, right? The panel 1, 2. And then because confocal is a 3D, right? So you can actually apply a 3-dimension of the synapse and then rotate. You can actually see the hexagonal structure that are very difficult to find if you're using EM, right? And of course, you can then try to use this resolution to measure the distance between the presynaptic protein versus the postsynaptic protein, and then you can measure the distribution of those protein distance in different brain regions. And you can also detect serial structure, ultrastructure, right? I mean, of course, you cannot compete with EM, right, in terms of resolution, but at least you can still see the nifold symmetry of the microtubulin bundles, in the real, and also, of course, also center singlet centroid. And then the advantage of Magnify or expansion is you can actually do a 3D easily, right, using this confocal microscope. You can even generate the whole, image the whole serial machine, right? From the basal body, the distal appendage all the way to the primary cilium, right? All the way. But still be able to resolve those structure. And of course, we try to do some demos, right? So we use Magnify. We expand different kinds of sample organoids with the cilia. And we have 3 different conditions: normal, drug-treated organoid, and also the organoid divided from the PCD patient. And then we do the statistics by counting the number of defective cilia. And then it turns out, we compare to EM results, they have similar population. So that means that there's no difference between using Magnify versus using EM to detect the ciliopathy. And Magnify can expand all kinds of different tissue type. And here we'll just show one of the example, the kidney disease, right? So renal pathology oftentimes require electron microscopy to detect and analyze features. For example, this is a podocyte. Looks like neurons. The processes serve as a filter. And then you have to look at those ultrafine structures. There are 200 nanometers in feature. And minimum change disease, for example, they're continuous. And you cannot detect the difference using optical microscope. But in our case, with Magnify, you will be able to see the advantage is because you're using conventional microscope, so you can actually still image the whole biopsy. And then dive all the way down to the podocyte ultrastructure. And with the pan-protein stain, which is inspired by one of the speakers' aunt's papers, and we can actually stain all the proteins using NHS Aster, and then give you an EM-like texture, right? The left is EM, the right is Magnify, but then you can also stain the immunofluorescence. So we have the ACTN4 that highlights the podocyte food processes. So it looks like the fingers, right? And then it looks like a clam, actually. We develop a pipeline, it's a deep learning, and then you can automatically segment and then annotate the food processes, nuclei, and basement membrane, which are the features that pathology care about. So we use this kind of pipeline. We are now collaborating with the local hospital. We're expanding 10 different kidney disease biopsies, and then we try to use this pipeline to determine whether they are diseased or not, and what disease, without any electron microscope, right? And you can also use this to expand pathogen-infected biopsies. In this case, so my student, Sherry, former student Sherry, actually developed this micro-magnify using a cocktail of enzyme to digest the cell wall from different pathogens, gram-negative, gram-positive, and fungal cell walls. In this case, we got the sample from Wills Eye Hospital in Philadelphia. They have this cornea samples that are infected by all kinds of different pathogens, right? Fungus, parasites, and bacteria. So we can actually expand them all and be able to identify, where the pathogen is and what's the morphology. Because Magnify kept all the epitopes, so you can actually do iterative immunostain. For example, in this case, we can stain 10 different biomolecules. With that, you can actually do a high-resolution co-localization analysis. You can generate a heat map. If you have different conditions, you will be able to be able to detect the difference, the changes between different proteins co-localization. If you're interested in doing Magnify protocols, you have two ways to do it. You can do DIY. We have all the resources available. You can just log in and then take a look at the white papers, the chemical sources, all that stuff. You will be able to buy them. We also have the JOVE video that you can see. But for a lot of people who doesn't want to spend time or money to do those things, we also try this easy-to-use kits. It's called Magnify Kits. We contains all the videos, just like what we did for the DIY version. But except that you don't need to worry about sourcing all the chemical. We do the QC for you, right? So basically, you can just get it, and then you can execute the experiment. At least you can try. And we have the demo box sitting in the sidecast table. If you're interested, you can take a look. So we already found that there are more than 150 labs doing these things. Again, we want to disseminate expansion microscopy technology. So this is one way that we try. Right? So now we already have more than 150 labs and biotech companies using Magnify all over the world. And that's one of the barriers. Another barrier is we try to screen the antibodies for you. So we convince the government to fund us to screen thousands of antibodies. And then we try this, and so far we already screened more than 400 commercially available antibodies, and we plan to screen 4,000 in 1 or 2 years. And eventually we'll publish this validated antibody database so everyone can benefit from this ever. We try to do workshops. One of them is, we recently did that in Cape Town. Actually, Carol Jacobs right here. It's great to see you. And we also do summer short course in some of the conference. Oh, wait a sec. Suddenly I realized, actually recently, that we are not the one actually doing the most magnified chemistry. It turns out E11 Bio, which is a focused research organization, they independently identify magnify as their chemistry. So they actually process like mouse brain, like 50 mouse brain per week to do these things and try to resolve the brain. I think this is something that's nice to see. You know, we hope that like more and more people will be able to use like expansion microscopy to actually like generate new technology and then make new discovery. That's what we want to see. And what's next, right? We try to expand size. Now we try to expand whole organ. So we are team up with Simon and Alan's in CBI, which is UPIT. And then we generate, like we build this metal beam, and then we try to expand the whole kidney, whole liver, you know, all kinds. And then, you know, generate this atlas-like database. And by the way, in order to do that, you will need petabyte servers, you know, sitting in the computer rooms to do this. For example, the silverfish, if you expand 11 times, it will take out like several petabytes of data, 3 months of imaging. So with that, obviously, I cannot do all this without all the amazing group members, as well as all the amazing collaborators. I'm always amazed by all the biological questions they bring to us, hoping that can be solved by expansion microscopy. And it's a great fun, actually. I learned a ton for that. And of course, eventually, my son did do the magnify and then expand the brain like 11 times. And thank you so much for the funding agency. Thank you.